Hello and welcome to NHM Live. I'm Alison. I'm your host for the very first episode of our brand new series. Now today we're asking who was Cheddar Man with Selina Brace and Chris Stringer. Now we are of course live in the studio, so this is your chance to put your own questions to our scientists. So please don't be shy. If you have a question during the feed, post that to us and we'll try to get through as many as we can. Now let's meet our first scientist. Selina, thank you so much for joining us on our brand new sofa today. You're the first person to grace our sofa.
Great.
So let's find out what you do here at the museum.
Okay, so I'm a researcher here at the Natural History Museum. I specialise in working with ancient DNA. So that's basically trying to get DNA from degraded museum and paleontological samples.
If it isn't a slightly daft question, what exactly qualifies as ancient DNA?
It's not a daft question at all. Ancient DNA is actually not so much anything to do with time. It's more about the conditions that a sample has been kept in. So ancient DNA we can describe as any DNA that was taken from a sample but not specifically stored or meant to be used for DNA analysis. So modern DNA would usually be taken from a live animal or individual and then immediately analysed, whereas ancient DNA is usually taken post-mortem, so from a dead animal, and then not stored for that purpose, and then we analyse it later.
And what sorts of specimens do you work with? Is it just human material? Is it animals?
Oh yeah, no, I work with lots of different types of materials, so it can be bones, hair, teeth, but from lots of different animals as well. So I've worked with insects, but small mammals, large mammals, and of course humans.
Fantastic stuff. Now, you have been involved recently in working on a project to sequence the DNA of quite a famous specimen here in the UK, Cheddar Man.
Yep.
So we're going to find out a bit more about that work throughout our broadcast. But before we do, let's find out a bit about Cheddar Man himself.
So I'm here with Chris and we're in the Human Evolution Gallery and we've come today to meet the Cheddar Man. So Chris, can you tell us a little bit about where Cheddar Man was found and how old he was?
Okay, well, Cheddar Man, it's a skeleton found in Goff's Cave, which is in Cheddar Gorge in Somerset. And we know from radiocarbon dating that that skeleton is about 10,000 years old. So it comes from the beginning of the warm period that we live in now.
So you see he was in a cave. Do you know how he got into the cave?
We don't know how he got into the cave, whether he's a burial, whether it's an accidental find. We don't have any real evidence of why he's in the cave.
So we can see here he is, the man himself, and we've got a fantastic skeleton there. Do we know anything about his sort of wider circumstances? Was anything found with the skeleton that was of interest?
There's a lot of material from the cave that's nearly 5,000 years older, but sadly from the time of Cheddar Man we don't have any definite artifacts. We know that people were putting their dead into caves at that time. It's possible that's what happened to him. And of course he was a hunter-gatherer, so these were people that were living off the land. This was before farming had arrived in Britain. So he's a young adult, we think, around 20 years of age. We don't know how he died, so there's a lot we don't know about Cheddar Man still.
And certainly he's a specimen. We've got lots of different specimens in the museum collection, but Cheddar Man is one of our sort of most special. Why is he such a significant specimen in our collection?
Well, it's very rare to have such a complete specimen from such time. You know, he's by far the most complete specimen from this time period and older in Britain. So in terms of the information we can get from the skeleton, it's important. But also, of course, important because we've got the DNA from Cheddar Man. We've got the whole genome of this individual who died 10,000 years ago.
Fantastic. And being that he's called Cheddar Man, would he have enjoyed some cheese, do you reckon?
As far as we know, there was no cheese in Britain. This was before farming had arrived in Britain, so sadly we can't associate him with any cheese.
Oh, well, there you go. Thanks very much, Chris.
That's all right.
Welcome back. And we're also joined on the sofa today by Chris Stringer. Chris, thank you so much for coming down and for that fantastic introduction to Cheddar Man as well. How long have you been working with this specimen?
Well, I think I first looked at Cheddar Man scientifically back in about 1970, so it's nearly 50 years, scarily.
Yeah, it's a long time. Yeah. Now, there's quite a lot that we can learn from the skeleton itself. So what sorts of conventional techniques have been used to study him?
Yeah, so obviously many people have measured the Cheddar Man skull, measured the teeth and so on using conventional measuring instruments. Nowadays we can use 3D scanners to capture the shape much more accurately. In the old days people x-rayed Cheddar Man to look at the internal anatomy. Now of course we can CT scan the material and really get a lot of detail of the whole skeleton and capture the whole individual.
And quite a healthy individual.
Yes, we don't know how he died, but he was certainly in pretty good shape. His teeth were in good shape. He's not very strongly built for his time, so doesn't look like he had a very stressful existence before he died of unknown causes.
Now, we unfortunately didn't have much material found with the skeleton itself, did we?
No, that's right. So this was excavated back in 1903, and it wasn't excavated scientifically. So they did save some material from around the burial, if it was a burial, the skeleton, but we don't know what really was associated. The material in the cave mostly seems to be from about 5,000 years older. So we've got human remains from an earlier occupation. For Cheddar Man, we've got no definite artefacts that seem to belong with him or his time period.
But we can infer from other finds, can we, from that era?
Yes, so there are thousands of Mesolithic sites in Britain, so this is the Middle Stone Age. And at that time, people were hunter-gatherers. We've got sites up in Yorkshire, for example, where people were living near a lake, and you've got beautiful preservation there, not only of stone tools, but also objects made of wood, and these beautiful artefacts made out of deer antler. So harpoons, spears, people were also using bows and arrows.
So we do know a little bit about how he might have lived.
That's right, yeah.
Now, of course, the newest DNA techniques are helping us to move Cheddar Man's story along, aren't they? We've, for the first time, sequenced his DNA. How is this now possible?
Well, the way we sequence DNA has actually sort of changed quite drastically over the last 10 or so years. We've gone from being able to target very small regions of the genome, usually like the mitochondrial genome, just a few hundred base pairs using techniques called PCR-based techniques. And now we have something called next generation sequencing, which essentially allows us to sequence all of the DNA that's in a sample. So we don't have to target just the mitochondrial or small regions, we can sequence it all, and it's actually a lot cheaper, relatively cheaper cost for us to do so.
Typically, how long does DNA preserve after death, and what sorts of factors would affect that?
So how long DNA is preserved is very much dependent on the environment that it's left in, or how it's buried, or how it's maintained afterwards. So immediately any organism dies, the DNA will begin to break down, but that's from enzymes and from microorganisms. And this is a process that is then followed by chemical breakdowns, so with like oxidatives, so oxygen and water. They break down the DNA and they cause bonds to break apart. So these things happen through time, but then we also, these are either sort of speeded up or slowed down dependent on the environment. So things like temperature, salinity, pH, humidity, all of that has an extensive effect on how quickly that DNA fragments and becomes into smaller and smaller pieces.
So what sort of environment helps to preserve DNA?
Really, you want a very cold environment. Permafrost is where the oldest DNA is always from, so temperature is a vital role. But constant is also very important, and dry as well. Those are what you really want.
So 10,000 years, obviously, it sounds like a very long time, but we do have genomes from older individuals, don't we? For example, Chris, we've got the Neanderthal genome, don't we?
Yes, that's right. So Neanderthal fossils, 40 or 50,000 years old, have given up their genomes. And in fact, there's early Neanderthal material from Spain, from the Cima del Huesos at Atapuerca. That material is probably at least 400,000 years old, and that has produced partial genomes of some of those individuals. So really it can be recovered much further back than we used to think.
And what's the oldest specimen that we've recovered DNA from?
The oldest bone specimen that's sort of well dated is actually from a horse, again from the permafrost, as I was saying, and that was about 700,000 years old.
It's very impressive, isn't it? Now, we've got some questions coming in from our viewers. So David on Periscope is asking, is it likely that Cheddar Man was part of a tribe or a group?
Yes, almost certainly. So looking at sites like the one Yorkshire Star Car, obviously there was a pretty big community there, at least seasonally. So they aggregated in larger numbers maybe in the spring and early summer there, and maybe then other times they would have scattered more widely. But yes, Cheddar Man would certainly have been part of a social group. And of course what we don't know is whether he was put in there intentionally, did he end up there by accident. But just down the road from Cheddar Gorge, there's the site of Avelin's Hole, and there possibly up to 50 skeletons from about the same age as Cheddar Man were put into that site. Sadly, many of them have been lost, but that shows us that sometimes some of these sites were used for many burials over quite a period of time.
And Mike on Facebook is asking, what was he doing in a cave full of artefacts that were 5,000 years older than him? There were previous occupants in that cave, weren't there?
Yes, that's right. So from the end of the last ice age, we've got a collection of individuals, and they seem to have been cannibalised. They've been eaten, and their bones date from nearly 15,000 years old. So that was an occupation towards the end of the last ice age. Then we have a gap with no evidence of human occupation that we know of until we get to this isolated discovery of Cheddar Man, nearly 5,000 years younger.
And Katerina on Facebook is asking, how old was Cheddar Man when he died, approximately? Do we know?
Yeah, so we think he was a young adult, so his third molars are fully erupted and developed, but they're hardly worn, so we would guess probably around 20. So quite a young adult, and we can say he's male, both from the skeleton and now from the DNA.
And of course, sequencing his DNA, that's quite a task. It's quite a complicated process. Now, Selena took me down to the ancient DNA lab a little earlier to talk through how we actually extracted the DNA. So let's take a look at that. So Selena, our ancient DNA lab is just behind you here. Now, we can't go inside today, can we? Why is that?
Yeah, sorry about that. But this is our very special ancient DNA lab that we have in the museum, and it's special mainly because it's super, super clean. As you can see, it's like an ordinary lab, but it's super clean in that everything that comes into the lab must be bleached, and every person that goes into there must be covered from head to toe in a hazmat suit, in glove, face masks, face covering, all of that, because basically in an ancient DNA lab you're trying to protect the ancient DNA, the fragmented DNA, from modern DNA. So we are walking pieces of contamination. We would be shedding our DNA all over the place, which would then interfere with our experiments, and it would mean we would be sequencing ourselves instead of Cheddar Man.
You can talk us through the process of how you actually extracted DNA from Cheddar Man. So how did you go about doing that?
Okay, yeah, so the first thing that you want to do when you're extracting DNA from a sample is to actually remove some of the bone powder from that sample. Quite often, as was the case with Cheddar Man, we actually have like a complete skull. So I've got an example of a skull here, if I can just show you. This isn't Cheddar Man, he's far too precious for me to be holding here. This is actually just a replica, so plastic. But what we want to do when we sample, when you've got a whole skeleton, a whole skull like we have here, is to go to the place where we would find the densest bone, because this means that the DNA is better preserved in a dense bone. And the densest bone that we know of is a bone called the petrous bone. This is actually an inner ear bone. So on a skull like this one here, you would see that would be where his ear would be there. If you turn him over, then you look inside, this bone here, this is the petrous bone. And so our first thing that we want to do when we're extracting DNA from a sample is to remove a very small amount of bone powder from this particularly dense bone that preserves the DNA inside of it. So what I would do is I would be inside this lab, in fact inside this hood here, because it's an enclosed area. I would take my drill, like this little drill here, and drill a very small hole up inside the petrous bone here. As you drill in through that one hole, then the bone powder just sort of trickles out onto the side, and it's that we remove and then begin our DNA analyses. As you can see with this drill, this is a very, very small drill. We have a very small drill bit on the end because we're trying to minimize any damage that comes to these precious samples that we have here in the museum.
And how much powder do you actually need? Is it not very much?
No, it's very, very little. Actually, we can work with about 15 milligrams. I mean, 20 milligrams is nice, but just to sort of an example, so 12 milligrams probably would fit in my fingernail. It's
Wow.
that small amount of bone powder that we remove.
So that's how we actually extracted the DNA. Now we've got a great question from Sylvia on Facebook saying, I think these bones are obviously quite fragile. How do we how do we balance the needs to analyse the specimen with the need to preserve it for future generations?
Yeah, that's a really good question. Well, we work very hard to be minimally invasive, as you saw from that clip there. It is one of the things that we've worked really hard over the years in is, you know, to make sure that we make a very, very small hole in any of the samples that we do. We try and use it and drill in places that are minimally invasive and don't have any effect on any of like the morphological aspects of it. We also try and use bones where you've got two of them. So like with the Petrus, you've got two of those, so you would just put a hole in one. So it is a balancing act, but one, obviously, we work in the museum that we take very seriously. It's an important point. Yeah.
Of course,
Yeah.
of course. You've got a very steady hand. Is it quite nerve-wracking working on some of these precious specimens?
Yeah, I know. When it's a really like iconic one like Cheddar Man, you're quite nervous when you go in. But you know, I've drawn a lot of these bones. So once you start doing it, to be honest, you're just thinking about drilling and you're thinking about getting the right amount of bone powder from the right place, and it's kind of calms you down a bit once you start it.
Now, we obviously extracted the DNA here. Did we sequence it at the museum as well?
Yes, we did. Yeah, so we've got a sequencing facility here in the museum. We have sequencing NextSeq machines that we actually use to sequence Cheddar Man. Yeah.
Very lucky to have all these facilities.
Oh, yes, we are.
And what sort of condition was his DNA in? Was it very fragmented?
Actually, for his age, Cheddar Man was actually pretty good. So we looked at a number of Mesolithic individuals, so from a similar kind of time point, and most of them had very, very little DNA. But Cheddar Man, very good. He had what we say is endogenous DNA content was 30%, and by that I mean that 30% of the DNA belonged to him, rather than being from like the soil bacteria and the other sort of extracted DNA that you tend to find in older samples. So yeah, he was actually very well preserved. Yeah.
Fantastic. Now, once you've got his DNA sequences, how do you then begin to interpret them? What do you compare them to?
Well, initially we would align the sequences to a reference genome, so a human reference genome that we know what the order is. It's a bit like a jigsaw puzzle in some senses, but in a straight line. You're trying to get the order of these fragments back in the right place, so you have like a reference to align them to. And then after that, we then would compare his DNA to other sequenced individuals. So these might be older individuals, so other Mesolithic individuals, or more recent ancient individuals that have been sequenced as part of other projects. And then you compare the changes that you see. So there are some base positions where, you know, I might have a C, you might have a G, and it's those variable positions that we focus on. They're called SNPs, so single nucleotide polymorphisms. We look at the differences, and that gives us an idea of the picture of populations and structures and what he looked like compared to other individuals.
Wow, exactly. That's one thing that the DNA can help us build a picture of is what Cheddar Man might have actually looked like.
That's
now,
correct.
using both a scan of Cheddar Man's skull and also the DNA data, the team did work with two brilliant model makers, didn't they, to create a reconstruction of what we think Cheddar Man might have looked like. And I think we've got an image that we can show. How did it feel seeing him for the first time?
Yeah, it was remarkable because there had been other reconstructions done of Cheddar Man. This one looked quite different, and the wonderful thing with the Kennis brothers is they really put character in their reconstruction. This is like a real person. Of course, it's only what he might have looked like. We don't know for sure, of course, what he looked like, but I think this really brings him home as an individual from that time period.
It really does. It makes such a difference, doesn't it? Being able to look into the eyes of an ancient relative. It's really compelling. Selina, having sequenced his DNA, was it?
Oh yeah,
Was it great to see him?
It was. It was really exciting to see him. I thought the Kennesis has done an excellent job, and I mean, for me, it was particularly nice to see him as kind of like a character, having only really seen him in a test tube prior to that. It was really nice to see him with character.
Yes. All the bones laid out.
Yeah, exactly. Mainly I saw him in a test tube.
Yeah, that's right. Yeah.
Putting flesh on those bones, very important.
It was.
Now, the DNA data suggested that Cheddar Man probably had blue eyes and darker skin. How surprised were you by that finding?
So in terms of the skin pigmentation is actually a very complicated trait to be looking at. Other individuals from this time period have been sequenced, like across Europe, and when people looked at their DNA, they found that they had some of these darker alleles or sort of variants that would usually indicate a darker skin pigmentation. So in that sense, in some ways, it wasn't that surprising because we had seen this in other individuals. I think what was interesting about this case was that we tried to take a little step further by using a published prediction tool. So this was a very recent set of more parts of the genome that you could look at essentially to tell this, and by using this tool we're able to kind of like put more of an image on what that sort of colour, or more of a range on what the pigmentation would have been like. So I think that was what was interesting.
It's absolutely fascinating. Now, the lighter skin colour, why did humans evolve to have that in Northern Europe?
Are you going for that, Chris, or am I?
Go on
then.
Yeah. We can both join in.
That's probably because of the in obviously Northern Europe we don't have as much sunshine, unfortunately, here.
No, we definitely don't.
So UV light and how we absorb UV light is actually really important for things like vitamin D, and we think that with the lesser pigmentation it means you can absorb more of that UV light, the little that we have here, and that then helps us with our vitamin D production. So we think that's why.
And possibly Cheddar Man and his predecessors didn't need to evolve that lighter skin because their diet included a lot of vitamin D. So they didn't have that pressure on them that people, when they took up farming with a less varied diet, maybe did need that extra boost from the sunlight to make their vitamin D.
Now, as well as telling us what Cheddar Man might have looked like, the DNA can also help us work out who he might have been related to as well. So let's hear now from Ian Barnes on that. So Ian, what does the DNA tell us about Cheddar Man's origins, where he might have come from?
So we're still not completely clear about Cheddar Man's origins. What we do know is that he looks genetically very similar to some other individuals from northwestern Europe who've also had their genome sequenced. So, for example, an individual from northern Spain at the Lobrania site, and another individual from Luxembourg from a site called Lochbourg. In fact, Cheddar Man really looks very, very similar to the individual from Luxembourg, but they're almost interchangeable. But where that population actually originated isn't clear at the moment. We think, though, that they probably spread out from the Near East at the end of the last ice age and recolonised into northern Europe over the course of several thousand years.
Okay, so one thing we're definitely not saying is he's not African, is he? That's something that people have been asking.
No, certainly not. He has African ancestry, but no more than I do. So, yeah, we think that his ancestors probably came out of Africa, settled in the Near East for some time, and then at the end of the last ice age, that group of people would have migrated north, probably through the Balkans and out into the rest of Europe. But the picture isn't really very clear at the moment because we don't have enough skeletons from that time period to actually tell exactly how things happened.
So how does Cheddar Man relate to us modern Brits? Has he left much of a genetic legacy?
Well, again, that's another question that's a bit difficult to answer, and even more difficult to sort of make a general answer about. What we definitely know is that after Cheddar Man, so throughout the rest of the Mesolithic, the population probably declined somewhat in population size and was eventually replaced at the beginnings of the Neolithic when farmers, again from the Near East, moved into Britain. So that's about 6,000 years ago. And then we think there's another population replacement from another group moving across Europe from Eastern Europe around 4,000 years ago. That's the Beaker peoples who arrived at the end of the Neolithic and the beginning of the Bronze Age. So that's two very large-scale population replacements that happened after Cheddar Man died. So we don't think that there's much chance that there's any direct ancestry in Britain today that you can link all the way back to Cheddar Man. What we do know, though, is that throughout Europe, and including Britain, we still have a small amount of hunter-gatherer Mesolithic ancestry in our genomes. And that's probably about 10% for people in Britain. It's a little bit more in some parts of Europe and a little bit less in other parts. So most people can trace back ancestry to Cheddar Man's ancestors, but probably not any kind of direct line to Cheddar Man himself.
Welcome back. Now we're getting lots of questions through from our audience. David on Periscope is asking, are humans still evolving in the modern day now that we don't have to do things like hunt for our food?
Well, certainly, yes, evolution is still happening. We are changing generation to generation, and there is dispute. Some people believe that with medicine and the way we live, that we're mostly shielded from the effects of evolution. But I would say that that's not true, because each generation we have maybe 50 mutations in our DNA compared with our parents. So generation by generation, that change is going on.
That's a great question. Great question, David. Now, we are sadly running out of time, but one final question for both of you. What do you think is the future of ancient DNA for unravelling our past?
Well, I think for the fossil record, it's going to continue to be really important. I mean, it's discovered an unknown kind of human. These people, the Denisovans that lived in Siberia, you know, they were tiny fragmentary fossils, and now we've got a whole genome of a new kind of human called the Denisovan. So I think there will continue to be revelations in the fossils giving us completely new perspectives. The interbreeding with modern humans, that had been debated for many years. The DNA finally showed it had happened. So I think that rate of discovery is going to carry on, and in the later time period, in Cheddar Man's time and more recently, even more so there, because the number of specimens that will give DNA is going to be greater and greater.
Yeah, it's an exciting time. Are there any particular specimens you wish you had the DNA from?
Well, personally, it would be great to get some more British ones that are older than Cheddar Man. 10,000 years is good, but we do have some older fossils. Wouldn't it be wonderful to have DNA from some of those?
Yeah. Oh, here's hoping, here's hoping.
Yeah.
Well, thank you both so much for coming to talk to us today. It's been an absolute pleasure. And thank you to our viewers for all of your excellent questions. Now, if we didn't get to your question, Chris and Selena will still be online for the next 10 minutes or so, so we'll try and answer the ones that we didn't get to. And we will be here again next month. We'll be talking dinosaurs, so do join us again. But for now, thank you and goodbye.